Okay, so this is our third video of this week, and this is the introduction to protein synthesis. So having finished the question pack now for topic 13, mass transport in plants, we're preparing to move on to topic 14. But before we get there, we need to recap quite a lot of prior knowledge that we've done since the beginning of the academic year on DNA structure, RNA structure, and also some of your GCSE that we can recap on protein synthesis before we start the new A-level content. Okay, so you will need a pen and paper. There's lots of tasks in this one, and you're going to be running alongside this, if you like, with the PowerPoint introduction to protein synthesis. You can kind of check again against that when you're revising as well. Okay, let's get going. Okay, so in this recap, I'm looking to review your DNA and RNA knowledge, introduce a few new key terms from the beginning of your student notes. So you will need some reference to your new topic 14 student notes that I've attached to your email. And then hopefully we'll get to the point where you can kind of see an overview and recap your GCSE knowledge, help you see the bigger picture as we move forward to doing this topic in full next week. Okay, so first things first, you need a pen and paper and you need to complete this task. So pause here. Okay, so you should be ready now to compare and contrast your prokaryotic and eukaryotic structure of DNA. Okay, so what have you got for prokaryotes? So for our prokaryotes, its prokaryotic DNA, it's short, okay, and found in the cytoplasm. It's circular and not associated with proteins, so no histones, and it doesn't contain introns. In contrast, when we think about the DNA of eukaryotic cells, okay, then it is very long, found in the nucleus, it's linear, it is associated with proteins called histones, and contains introns. So you'll find this table at the beginning of your topic 14 keynotes as a recap from your cells topic. So in your cells topic, we ask you to compare and contrast prokaryotic and eukaryotic structure. Within there, you should have these bullet points for DNA. So we're moving on to protein synthesis. And within that topic, we will need to know about the differences and similarities in DNA in eukaryotic and prokaryotic cells. Okay, so note here, in your keynotes, it says prokaryotic cells DNA is the same for DNA in the mitochondria and the chloroplast. Now you'll remember the reason for that is that we believe prokaryotic cells are the evolutionary origins of mitochondria and chloroplast, so they used to be bacteria. And therefore they contain DNA, and the DNA they contain has all these features. That's evidence we believe they were once prokaryotic cells. So flip it and give you a different question. On the new spec, even the AO1 questions require a little pondering before you hit the answer. So imagine we were asked to give 3 ways in which mitochondrial DNA differs from the DNA found in the nucleus in a eukaryotic cell. At first we might think, oh, we don't know the answer, but then we'll remember that mitochondria have DNA that is prokaryotic in its features. So we'll get our mark points for giving all the differences between prokaryotic DNA and eukaryotic DNA. So let's have a look at this student response. How well do you think they've done? They've said DNA molecules in the nucleus are very long, linear, and associated with histones, whereas the mitochondrial DNA is short, circular, and not associated with histones. So actually, they've done a pretty good job of picking 1, 2, 3 things that they have contrasted that are different. So they would get they've been very concise, but they get 1 mark for long versus short, 1 mark for linear versus circular, and 1 mark for associated with proteins versus not associated with proteins, or not associated with histones, the name of the protein. Okay, moving on to some key terms. So in your keynotes now, you will see that there's some extra key terms before we review and recap DNA and RNA structure. Okay, so the first key term to make sure we fully understand is the word genome. Okay, so in your keynotes, this is the exam board definition. It's the complete set of genes in a cell. Okay, that's what they say. So your genome, okay, is all your genes present in your cell. So that would be for every single chromosome that makes up your genome, every single gene. But that's the exam board definition. But then they expect you to note this as well. So you might want to add this to your keynotes here. So although genome is the complete set of genes, included in genes is not just the coding information. So there is a lot of non-coding information like introns in eukaryotic cells that also makes up an organism's genome. Okay, so complete set of genes in the cell, yes, but it is basically all the DNA base sequence for that organism, including non-coding information. Contrast this with the proteasome. Okay, so the exam board's really keen on this. They've already asked this one for a 1 mark definition. So what they wanted was that it's the full range of proteins a cell is able to produce. And when you look into this, the comparison between an organism's genome and proteasome, you'll find that they're very different. So we've got about 21,000 genes. Okay, but when you look at how many different types of proteins we can make from that coding information, it's a lot bigger. So that's because, of course, a polypeptide chain, a protein might be made up of more than one different combinations of polypeptides. There might be some modification after translation where a bit's cut off or a bit's joined on. So by the time you've finished, we've got a much greater variation of proteins in the proteasome. Okay, so interesting question now. How is determining the genome of a simpler organism helpful in vaccine design? So this is a link to year 2. Next year we're going to do gene expression, and we'll come back to the genome and we'll talk about the Human Genome Project, which was the mapping of all the genes on all the chromosomes for a human being. So we got the full complete set of genes mapped out in the human genome. Okay, so think about simpler organisms, for example our COVID-19 virus. At the moment there's a lot of people trying to design a vaccine that's going to work to protect us from this virus. So how does determining the genome help us? Well, simpler organisms are simple because their genome basically codes for their proteome. So all of the genetic information inside a virus basically codes for a protein or viral RNA or DNA. So we can start with that coding information, the genome, and we can work out things like antigen proteins from that, which can help us then to target them in vaccination. Okay, so I had a quick toot on the internet and someone's already deciphered the genome of COVID-19, and on the internet, free to all scientists to use, are all the different genes that have been mapped. And now scientists can look at each gene and see whether they think it might be a novel protein that could be an antigen and a valid vaccine target. So that's where they're getting their starting point. Okay, so why is it much more difficult to understand the genome sequence of a more complicated organism? So again, this is a link to year 2. Like ourselves, we're very complicated. So in our genome, we've got genes that code for the proteins, the proteome, but we've also got a whole load of non-coding DNA. We've got introns that you know about already. We've also got things called variable number tandem repeats, promoter regions. We've got termination regions as well that we'll learn about in year 2. None of those code for amino acid sequences in proteins. So it's very difficult to work out what does code for a protein and what is part of this non-coding genome, making it very difficult to interpret human genome information. Not impossible though, okay, but just takes a little bit longer. Another thing to put into the factor there is just to show you how many different genes for different organisms there are. So when you get to a complex organism like us, you think we'd have the most number of genes, but we don't. There's other organisms with a greater number of genes in their genome. They'll also have non-coding information in there as well. Okay, coming back to our key terms. Chromosome, we should know, you should be able to define this one. So a DNA molecule and its associated proteins. So there's our DNA molecule wrapped around histones. That condensed form is our chromosome. Remember, following DNA replication, that will be duplicated. So just a reminder here that following DNA replication, we'd have 2 sister chromatids making up the chromosome. What's a sister chromatid then? What's a chromatid? Well, it's one of the strands of DNA which make up the chromosome. Just at that point, following the S phase, following G2, when we move into prophase, prophase 1 in meiosis, where we've got 2 copies of the DNA molecule, 2 sister chromatids joined by a central centromere, okay, just prior to cell division. Obviously, we call a chromosome, when it's 2 sister chromatids, a chromosome, but when it's just one DNA molecule on its own, when we're in histones, that's still a chromosome. So when that sister chromatid separates during cell division, we still refer to the individual chromatids as chromosomes at the end of the process. Okay. Gene, another definition we should know. It's the base sequence of a DNA that codes for an amino acid sequence of a polypeptide or a functional RNA. So a functional RNA might be a tRNA or an rRNA, as we'll see a little bit later on. Okay, locus. Again, this is a key term that we will use a lot. We'll use it a lot in our genetics next year. So it's our introduction to this key term. So the word locus or loci is the location of a gene on the chromosome itself. So here we can see in this diagram, this is the genetic locus for the gene labeled R, and it's found at the same position on both homologous chromosomes, so both on the chromosome inherited from the father and the chromosome inherited from the mother, this being a diploid organism. Okay, so you'll find that gene on the same chromosome at the same locus. That's why it's the same gene. But it doesn't have to be the same form of that gene. So remember, we need an allele definition as well. So an allele is a different version of the gene. Okay, so if we go to this one here, gene C, you can see that this individual has inherited a recessive C from the father on this homologous chromosome and a dominant C from the mother on the other homologous chromosome. So they're heterozygous for that characteristic, which means that at that position on that DNA molecule, the base sequence for the recessive gene is a little bit different to the base sequence for the dominant gene. Okay, so the actual base sequence would differ at that position. Remember, this is going to be key to our genetics understanding, one of our topics of year 2, if that's one of the things you're really interested in. Okay, so there are key terms. Of those key terms then, here's 2. Can you now write down your definitions? And then did you pick up what an intron is? Have a go. Pause now. Right, you should have rejoined us. So gene, that was the base sequence of DNA that codes for the amino acid sequence of a polypeptide or a functional RNA. And you need to have that functional RNA there. 2, the locus. Okay, that's the fixed position of a gene found on a particular DNA molecule. So the fixed position of a gene on a chromosome. I'm just going back to that definition. So that's a DNA molecule, a chromosome wrapped around histones, isn't it? And finally, an intron then. So introns are non-coding base information, and as we'll see, these are actually found within a gene. Okay, so there's non-coding information found between genes, but there's also non-coding base sequence information found in the middle of genes as well for eukaryotic cells. So if you've got at the moment that introns are non-coding information in genes, brilliant. Okay, so we're on to recapping our DNA and RNA structure and function. You have got keynotes now which are recap as you go through the keynotes. So hopefully you'll remember all this information from and be able to do these tasks, but if not, you can always have a sneaky peek in those topic 14 student notes at the start of the topic. Okay, so DNA and RNA, what do we know? What's the role of DNA in a living cell? Write down your answer. Okay, we should have that it holds the genetic information. So it holds the base sequence for the codes for proteins and functional RNA. So it's where we find our genes, our genetic information. What's the role of the mRNA? Write it down. So we should have mRNA transfers genetic information from DNA to ribosomes. So that's what its job is. It takes the gene information, so a copy of a gene, from the DNA to the ribosomes, as we'll see later on in the topic. What about tRNA then? Do you remember? Its job in this whole process of protein synthesis is it carries a specific amino acid to the site of protein synthesis. So here's a kind of overview of the process we're going to be learning about in topic 14. Here's our DNA with our genetic information. So here we've got the base sequence that codes for, it could be a protein or a functional RNA. In the process of protein synthesis, a transcript, a copy of this, is made in the form of messenger RNA that can carry that genetic information then to the ribosome, where it is read using tRNAs that carry specific amino acids to the site, which are then joined together in the specific order that is read from our mRNA. Okay, so this first part we call transcription, and that's where we make our mRNA molecule. And this second part is called translation, and that's when we convert the code in the mRNA base sequence to an amino acid sequence using the ribosome, the tRNA, and the mRNA as a template. Okay, so what do we know about structure then? DNA and RNA both are polymers, and they're produced by joining which monomer type? Okay, get yourself drawing now. I'm hoping we've recalled it's a nucleotide. Okay, so both DNA and RNA are nucleotides, but can you draw and label one for me? Generic structure. Okay. Should look like that. We should have a pentose sugar, phosphate group, and a nitrogen containing base. Now, depending on whether you're going to build DNA or RNA, that nucleotide will be slightly different in terms of its sugar and the bases it contains. So how? What would be the 3 components if it was a DNA nucleotide? Okay, would have a phosphate. The sugar would be deoxyribose, and there would be a nitrogen containing base. Now those nitrogen containing bases of 1 of 4, okay, could be that it would be adenine or thymine, could be guanine or cytosine. But if this was an RNA, then that would be different. The sugar would be ribose, and of those bases, there's no thymine in RNA nucleotides. In DNA nucleotides, the bases guanine, cytosine, adenine and thymine are found. But in contrast, in an RNA nucleotide, it's guanine, cytosine, adenine, but uracil in place of thymine. So can you use that now to answer this question? Describe how a DNA and RNA nucleotide differ. Pause and answer. Okay, so we should have a difference in sugar. So DNA nucleotide contains deoxyribose, RNA nucleotides contain ribose, not ribosome. That is not meant to say that. Giving the game away on the rest of it. Okay, so DNA nucleotides contain organic bases adenine, cytosine, guanine, or thymine, whereas RNA nucleotides contain adenine, cytosine, guanine, but uracil instead of thymine. Tick, tick. Okay. What type of bond forms then when you join nucleotides by condensation reaction, whether you're making a DNA molecule, joining DNA nucleotides, or making an RNA, joining RNA nucleotides? It should be a phosphodiester bond, yes. Okay, now thinking about DNA then, it's double-stranded. I'm saying mainly because you're going to meet 2 single-stranded DNAs in year 2. But for the moment, if we just accept most of it is double-stranded, unless they tell you opposite, you're going to assume it's double-stranded. You have base pairing in DNA, a base pairing set of rules as well, which you don't have for mRNA. There's no base pairing in mRNA at all. Okay, there is some base pairing in your other RNAs, but they are not double-stranded, so don't obey the rule. Okay, so rules about base pairing then are, we know what our bases would be. So can you name 1 to 3 from the information provided? Okay, pause, answer, and then join again. Should have thymine, because it is the base pair that binds to adenine. Adenine is the double ringed, so that's why it's got a bigger box. Single ring for our thymine, so it's shown with a smaller box. Then in terms of colours, 2 different colours, different shapes as well, to show us this is the other base pair. So other base pair, the bigger box would be the double ring, so guanine, and the single ring then for cytosine. So our single rings, pyridamines, and our double ring, bigger purines. Okay, what type of bond then would be found between complementary base pairs within a DNA molecule? Hydrogen bonds. Super. How many? Well, depending on the base pair. So between adenine and thymine, we would expect to have How many? Okay. And how many between cytosine and guanine? Okay, if you're not sure, if you go back to that picture, the game is given away there. So between guanine and cytosine, it's 3 hydrogen bonds. Between adenine and thymine, 2. Okay, so one thing we can do with double-stranded DNA is we can predict the percentage of each base from just one figure. So, can you have a go at that? I'll give you the answer. How would you work it out? So it's percentage, so they should all add up to 100%. If they are the same base pair, they should be equal. So adenine and thymine would be 35. Add together, make 70. Take away from 100 gives you 30, and then split it for each guanine and cytosine. Okay, so let's see if you remember that definition. What is an allele? Okay, you should have an allele is a different version of a gene. I tried to show you that by giving you some different alleles here. They differ in base sequence from each other. She says, there and there. Okay. Right. So the last thing we need to recap is our RNA. So we know there are 3 different types of RNA. Okay, so we might be asked to give examples of functional RNA. Of these, when they're saying functional RNA in that gene description, that gene definition, so a base sequence that codes for the amino acid sequence of a polypeptide chain, or the base sequence of a functional RNA, we're talking about tRNAs and rRNAs. Okay, so we've got messenger RNA, transfer RNA, and ribosomal RNA. We need to know a bit more about the structure of messenger and transfer, and then after that, really all we need to know about ribosomal RNA is that it is combined with proteins to form the large and small subunits of the ribosome. So this is stolen from a textbook. So you can see messenger RNA, you find it in the nucleus where it's made, and then it migrates to the ribosomes in the cytoplasm where it's used, and it carries the DNA sequence information to the ribosome. The transfer RNA is in the cytoplasm. It provides a link between the mRNA and the amino acids, so it transfers the specific right amino acid to the ribosome to the right position. Okay, and then finally ribosomal RNA in the cytoplasm, and it's a structural component of the ribosomes. So for RNA, messenger RNA, can you have a go now at describing the structure and then try and relate that to its function? You can use your keynotes. Pause and then replay. Okay, so it's a single-stranded polynucleotide. The idea it's made from RNA nucleotides. It's a copy of the base sequence of a gene. That's a description of its structure. How does that relate to its function? Well, it's single-stranded. That allows tRNAs to bind to it. That's really important to its function, as we'll see next week. Also, each codon codes for one amino acid. Again, is an important link between its structure and function that you'll see next week. Okay, tRNA, describe the structure and relate to its function. A bit more description now. You can see the special regions to join an amino acid. There's a special region at the bottom, which is the anticodon, complementary to the codon on the mRNA. So have a go answering that one. Okay, you should have paused and restarted. So go through the answer. So again, it's a short single-stranded polynucleotide. Sometimes people don't get that, but if you look, it's one single strand that's folded back on itself in this clover shape. So it's made of RNA nucleotides. It's folded into a clover shape, and it has an amino acid binding site. So that's at the top there. And at the bottom, an anticodon. Anticodon there at the bottom. It contains an anticodon of bases at the bottom and the corresponding amino acid at the top. So it always brings the right amino acid to the right position on the mRNA. It only binds to the right position because it's got its complementary anticodon, and the amino acid it carries is specific to that anticodon. So it always brings the right one to the right codon, as we'll see next week. So don't worry too much if you need to develop the function bit on those answers yet. So our rRNA then, all you need to know about rRNA is it's combined with protein to form two subunits, the small and the larger subunit, that make up the ribosome. That's it. So focusing then on our mRNA and our tRNA, can you give two differences between their structure? Pause, have a go, and then rejoin. Okay, so mRNA is longer. Okay, it's got more nucleotides than tRNA. Now, sometimes students don't get that, but I'll take you back to this overview picture. Okay, the mRNA is one long single-stranded polynucleotide. The tRNAs are pretty small, can you see, in length, much smaller, much shorter than this long mRNA. So that's the first mark point, so sometimes people miss that one. Second one, the mRNA is a straight molecule, but tRNA is folded into that clover shape. I'm pretty sure you've got that one. mRNA contains no base pairs at all, so there's no hydrogen bonding, but the tRNA has got base pairing. So if I take you back to the picture of it, can you see here all the hydrogen bonds between complementary bases? So it's actual base pairing between the same strands. It's only single-stranded still, but there are these regions of complementary base pairing to hold that clover shape. Okay, so there are your 3 mark points, and that has been up as an exam question now, so you can see this is a new spec mark scheme. Okay, task, and you're going to have to pause on this one. Copy out the table and then complete it for DNA, mRNA and tRNA. Okay, so your answers for this are sugar for DNA, deoxyribose, but ribose for both of our RNAs. Bases, adenine, thymine, guanine, cytosine for our DNA, but adenine, uracil, guanine, cytosine for both RNAs. Number of strands, 2 for DNA, 1 for both RNAs. Size, very much the largest, longest, because contains many base sequences for many genes, is DNA. Short in comparison to DNA is the mRNA, because it only contains the base sequence for one gene. Yeah, so the base sequence for one polypeptide or one functional RNA. But by far the shortest are the tRNAs. The next one's stability, so how long it lasts. Okay, so DNA is very stable. As you know, we've been able to extract DNA from bone samples hundreds of thousand years old, or teeth, tooth enamel, or insects encased in amber. So it's a very stable molecule, mainly because of all that complementary base pairing between the bases, and the bases being on the inside of that double helix. But the opposite end of the spectrum really is our mRNA, which is actually degraded by the cell. Now you might think, why would you want to do that? But when you copy the gene into an mRNA molecule, the cell will keep making whatever protein that is, or whatever functional RNA is coded for by the RNA, the mRNA, until it's broken down. So you wouldn't want it to last for a long time. You'd rather remake it if you still need that protein or functional RNA. Okay, so that's less stable. The tRNAs are fairly stable though, again, because you've got that complement, so you've got some complementary base pairing in that single-stranded molecule, just making it more stable. And the final one, hydrogen bonds, yes for DNA, yes, but not as many obviously for tRNA, and none at all for mRNA. Okay, so that's where we're going to finish our recap for today. Okay, by all means go back through your PowerPoint and test your knowledge again just before you start next week on this topic. In the meantime, it might be worth checking out your GCSE resources from when you were at school. If I asked you to describe how the base sequence of a gene determines the function of a protein it codes for, how far do you think you'd get with that? What would you already know from GCSE? So that's your thinking point to end on in this video. Thank you. Ooh.